All right, thank you everyone for being here. Today I'm going to introduce you to the field of plant biochemistry. And this project was done in collaboration with Dr. Paul Larson in the Department of Biochemistry. So Arabidopsis thaliana is a biochemical model organism that is widely used in the plant biochemistry field. It's a dicotyledonous member of the mustard family, and it's been researched for decades in plant biochemistry, so its genome is very well known. And within the genome, there are genes that code for pectate lyses. And what these are enzymes that cleave pectin, which is a complex carbohydrate, and it cleaves it into oligogalacturonic acid fragments, which I will refer to as OGA fragments. So one of the mutants that we study is the Pmr6 mutant. So this mutant has a loss of function of the Pmr6 gene. So it has a loss of function of enzymatically degrading pectin into OGA. And the reason this mutant was first discovered was because it's naturally powdery mildew resistant. And in this image, you can see powdery mildew on the wild type here. And on different Pmr6 mutants, they are naturally powdery mildew resistant. So this is why it is a widely studied organism, the mutant specifically. And we see broad phenotypic changes in the mutant along with powdery mildew resistance. We see hypocotyl growth retardation. And on the next slide, I will show you what the hypocotyl is. And we also see alterations in the ethylene response pathway within the mutant. So the Pmr6 substrate is likely a cell wall derived oligogalacturonic acid. And we can confidently assume this because there are about 20 other pectate lyse genes that have been widely studied. So we very well understand the role of these pectate lyse genes in cleaving pectin. So we can assume that the substrate is an OGA. And because the mutant has the inability to enzymatically cleave the alpha-1,4 linkages in pectin, we can assume that the substrate is building up in the mutant. So we suspect that the OGA builds up due to an inability for enzymatic cleavage. And we believe that this OGA is having bioactive properties within the mutant, and that is what is causing the phenotypic changes in the mutant. Previously, it was suspected that powdery mildew resistance was due to strengthened cell walls, since the mutant has the inability to cleave pectin that would strengthen the cell walls, and they thought that powdery mildew resistance was due to that, but that has been proved incorrect. And because of that, we want to investigate why the mutant is powdery mildew resistant and how the OGA is influencing the mutant's phenotype. Ethylene is a plant hormone that is involved with the growth and senescence of plants. It's very important and is involved with all life cycle stages of the plant. And when seeds are grown in the light, ethylene causes the hypocotyl to elongate. And when seeds are grown in the dark, it causes the hypocotyls to shorten. So when we expose plants to Pmr6 substrate, we can cause their hypocotyl to change. So the hypocotyl is this region here. It's much like a primitive stem in the plant. Excuse me. And what we do is we take the Pmr6 mutant seedlings, we grow them, and we collect them, and we process them through a soluble sugar extraction process. And the goal of this is to manipulate biochemical aspects of sugars to pull out the OGA from the plant. Once we've extracted the substrate, we apply it to a growth media for wild type, so the normal plant, and for the mutant plant. And what the goal is to see how the substrate influences plant growth. We also add silver nitrate and AVG to the growth medium. And what these do is they knock out ethylene reception and biosynthesis, so that while we're measuring ethylene response, we're only measuring it in response to the OGA, not ethylene itself. And we grow the seedlings in dark for 4 days, and then we observe the lengths of the hypocotyl and compare them to the wild type and to light grown seedlings. So when ethylene is exposed to dark grown seedlings, the hypocotyl is retarded in length. So we've taken the Pmr6 substrate from the Pmr6 mutants. We've applied it to wild type and Pmr6 mutant plants to see how the substrate changes hypocotyl growth. And what we see is that we can induce hypocotyl shortening in the wild type plant. And we've also purified the Pmr6 substrate and shown that it is larger than 1 kilodalton. And we've also shown that the Pmr6 substrate displays heat stability. So what we did is we boiled the substrate and then applied it to plants, and we saw the same results, that the substrate still induces hypocotyl shortening even after it's boiled, which indicates that it is a carbohydrate that we are observing, not a protein, because protein would be denatured by high temperatures and the hypocotyl retardation would not be observed after boiling. So this supports our hypothesis that the Pmr6 substrate is a carbohydrate. Additionally, it was previously suspected that the reason why the mutant's hypocotyl does not elongate is because of the strengthened celled walls and an actual growth defect that prevents it from ever elongating. So what we did is we exposed the plants to ACC. ACC is a non-protein amino acid that is the metabolic precursor for ethylene. So via Le Chatelet's principle, we can induce the ACC and force the plants to make ethylene and force the ethylene response in the plants. And what we see from this is that the mutant is capable of elongating its hypocotyl to the same heights as the wild type plant. So this shows that there isn't a growth defect in the plant and that hypocotyl retardation in the mutant is due to some type of signaling pathway going on. Additionally, we see in the mutant that its roots grow similar to the wild type roots, which also indicates that there's not some general toxicity or general growth defect, but that the OGA is playing a bioactive role within the plant. It was also previously hypothesized that the mutant is naturally powdery mildew resistant due to amplification of defense gene expression. So we took wild type plants and we exposed them to Pmr6 substrate, and via RT-PCR, we observed defense gene expression, specifically the PDF family and the ERF family, which are the major defense gene components in Arabidopsis. And what we actually saw was diminished expression of PDF family genes. So this indicates that exposure of the wild type to the substrate depresses defense gene expression. So this was contrary to what was previously hypothesized. And this is really interesting because it opens up the door to what really is causing the powdery mildew resistance if it's not the defense gene pathways within the plants. So this is currently being investigated further, and the true cause of the powdery mildew resistance is still unknown and could have to do with the bioactive properties of the OGA in the mutant. Lastly, we wanted to see if the mutant, if the Pmr6 substrate is playing a role in the ethylene response pathway in the mutant independent on its own. So this could open up the door to a completely novel biochemical pathway that has been unknown to the plant biochemistry world by the OGA. So ETR1 is a gene that is necessary for Arabidopsis to sense and perceive and produce ethylene. So basically the entire ethylene response pathway requires this ETR1 gene. So we knocked out the ETR1 gene in wild type and mutant plants. So now those plants with the mutation are completely incapable of sensing, perceiving, responding, producing ethylene. So even if ethylene is present, we shouldn't get the response. We linked this mutation to an IN3 GFP marker. So what this means is essentially we know which plants carry the ETR1 mutation. We know which plants are ethylene insensitive. GFP is a marker that's commonly used in biochemistry. And what it is it glows when the gene is being expressed. So you can see that in this image here, these glowing spots are GFP expression. So the reason why we link GFP expression with IN3 is so that if the IN3 is being expressed, we can visibly see it by glowing. So what we did is the ETR1 mutation was applied. These plants are completely ethylene insensitive. Then we applied the substrate to the plants, and what we see is activation of the ethylene genes, even though there is a mutation in the ETR1. What this indicates is that the OGA is playing a role in the ethylene response pathway independent of the ethylene genes. So this either indicates that there's crosstalk between the OGA and the ethylene pathway, or that the OGA is acting in a novel pathway that has previously been unknown. And this will be further investigated by the Larson lab. So we want to further investigate the role the substrate is playing on ethylene signaling, either via crosstalk with the ethylene pathway or in a novel biochemical pathway. Additionally, we want to further refute the idea that the mutant has growth retardation issues and growth defects by applying auxin and inducing apical hook formation and hypocotyl elongation. So the mutant naturally doesn't elongate its hypocotyl and naturally doesn't form an apical hook. So by inducing auxin and causing those to form, we can show that the mutant is normal and capable of those things, but doesn't do them because of the substrate. We also want to continue purification and characterization of the substrate. I will now open the floor for any questions.